So estimation of total serum protein by Biuret method. So before that you did estimation of glucose by which method? Godpot method. Okay. So introduction, what is plasma protein? Blood contains large varieties of proteins. The major ones are albumin, globulin, fibrinogens. Other examples are proteoglycans, metalloproteins, lipoproteins, etc. Now proteins are synthesized mostly in the liver except immunoglobulins and hormones. Where are immunoglobulins synthesized?
Lymph nodes.
White blood cells. And hormones, which gland? Which gland? Hormones are synthesized from endocrine glands. Yes. So function of proteins are they act as enzymes, hormones, clotting factor, transport agents, protective agents, nutrient agents, immunity, and etc. So these are the various functions of the proteins. Proteins are synthesized in the liver. Okay, these are the various functions of the protein. Now, albumin helps in the maintenance of colloidal osmotic pressure. Who can tell me what is colloidal osmotic pressure?
On the capillaries.
Yes, pressure inside the blood vessels. So, our blood vessel has a certain type of pressure known as the colloidal osmotic pressure. Now, albumin helps in maintaining the colloidal osmotic pressure. Why? Because if we don't have the colloidal osmotic pressure, our blood vessels will collapse. If it collapses, if the blood vessel collapses, then the blood cells, the fluids and other substances will not have a free flow. If the blood vessel is supposed to be like that, means if it is like this, means it has the colloidal osmotic pressure. But if albumin is not helping in maintenance of that, then it will collapse. And how will the substances free flow? It can't, right? So albumin helps in the maintenance of colloidal osmotic pressure. Then we have maintenance of acid-base balance, that is the buffering action of protein. Now if your body has more H plus ions, what does it mean? Your body is acidic or basic? Acidic. Now if your body is having more H plus ion, means the proteins will go and bind with the H plus ions. Now in that process, the concentration of the H plus ion will decrease, right? It will decrease, right? If the protein goes and binds with them. So ultimately the concentration of the H plus ion will decrease. Similar wise, if your body is having more OH minus ions, means your body is in basic or alkaline state. Similar wise, your body, if it is having more OH minus ions, the proteins will go and bind with them, and in that process the concentration of OH minus ions will decrease, bringing your body into a normal state. So as when we say that our body has certain coping up mechanism like maintenance of the acid-base balance, so this is how it is happening. This is how, by the help of the buffering action of the protein, the acid-base balance of the body is maintained. Okay, so this is another function of protein. Then we have the transport mechanism, that is albumin, alpha globulin, beta globulin helps in the transport. So can you tell me how does bilirubin get transported from the blood to the liver? By binding with albumin. So that's how albumin helps in the transportation. Then defense mechanism, that is done by the gamma globulin. Now globulin is one of the major components of protein. Now globulin has other subtypes, that is alpha, beta and gamma globulin. Now gamma globulin here acts as a defense mechanism. How? It helps us fighting against infection. That is the defense mechanism of gamma globulin. Helps us to fight against infections, various infections. Then we have blood coagulation done by fibrinogen. Blood coagulation done by fibrinogen. Now fibrinogen is one of the clotting factor. There are 12 clotting factors. One of them is fibrinogen. So what happens is, when you get a cut, you start to bleed. But after some time the bleeding stops and you get some clotted dry blood on top of the cut. Right? Yes or no? Am I right? Yes. So what happens there? Why does the bleeding stop?
Temporary treatment.
Yes, so the fibrinogen goes and clots in that cut area. Along with that, other blood cells also goes and helps in formation of a clot. Okay, other blood cells like RBCs, WBCs, along with fibrinogen, they go in the cut and they stop the bleeding and forms a clot. Okay, then after that, the dry blood that you see, that is the fibrinogen and the blood cells. Okay, so that's how it helps in this blood coagulation. Then what is plasma protein? Plasma protein is albumin, globulin and fibrinogen combined is known as plasma protein. And another thing is, what is serum protein? Now first tell me what is serum. Centrifuging. Now which portion? After centrifuging, how will you get? What will happen to the blood inside the test tube? The upper supernatant liquid is known as the serum and the lower part where the sediments are there, the RBCs and the fibrinogen will settle down below. Why? Why is fibrinogen absent in serum? Why is it absent?
Precipitated.
It precipitated, yeah, but why? Why is it not there in the supernatant portion? Because it is a clotting factor, so it settles down. It gets clotted with the other substances in the blood and it settles down. Okay, and the supernatant portion is the serum. And what is serum protein? Serum protein is plasma protein minus fibrinogen. Plasma protein minus fibrinogen is serum protein. Okay? Okay, so the normal range. Total protein ranges between 6.5 to 8.5 gram per dl. In some books you can get 6 to 8. Both are correct. Then we have serum albumin ranging between 3.5 to 5 gram per dl. Serum globulin ranging between 2.5 to 3.5 gram per dl. Then we have fibrinogen ranging between 200 to 400 milligram per dl. Please don't confuse these two. Okay, this is milligram, these are in gram. Okay. Then albumin globulin ratio ranging between 1.2:1 to 2.5:1. If albumin is less in Ag ratio, what is the meaning of Ag ratio? Ag ratio means albumin divided by globulin. If albumin is less, your Ag ratio will be less. And if globulin is less, your Ag ratio will be more. Okay? And both are abnormal conditions. If it is less, that is also abnormal. If it is more, that is also abnormal. Okay? Then now the principle of Biuret method. All compounds containing two or more peptide bonds react with cupric ions in alkaline medium to form a purple color complex. Now what is protein? Is protein a single molecule? Is protein a single molecule? No. It has multiple amino acids. Now how does the amino acid bind with each other? In peptide bonds. So when there are two amino acids, how many bonds will be there? One peptide bonds. So will you get the test? Will you get the proper result of this test if you do? No. So you have to have two or more peptide bonds. Okay, two or more peptide bonds which will react with cupric ions in alkaline medium. Now this cupric ions and the alkaline medium will be provided by the Biuret reagent, in the Biuret reagent, to form a purple color complex. Now what was the color in the glucose estimation by Godpot method? What was the color? Pink. It was pink, and in this test you will get purple color. Okay. The absorbance of the complex is measured calorimetrically at 540 nanometer. Calorimetrically at 540 nanometer. Now the kit that you will be using, okay, it's a coral kit. Coral kit, the kit that you will be using inside, these are the coral kit. Now according to the coral kit, you should use 550 nanometer, not 540. Here I have put 540 because the calorimeter that we have, it does not have 550, it has still 540. And for glucose, how much did you use? 520. For this one you will use 540, but according to the kit you should use 550. But we don't have 550, that's why we will use at 540. Okay, so the reagents required. Biuret reagent, which consists of copper sulfate, which is the source of copper ions. Then sodium potassium tartarate, which stabilizes the copper ions. Now, so that there is no hyper reaction taking place, this sodium potassium tartarate stabilizes the copper ions, which was sourced by copper sulfate. Okay. Then we have potassium iodide, which acts as antioxidant. What is antioxidant? So that the oxidation does not take place. So potassium iodide acts as antioxidant. Then sodium hydroxide provides the alkaline medium. In your Rothera's test, what provided the alkaline medium? Liquor ammonia. Don't get confused between the two during your viva. In Biuret, this alkaline medium is provided by sodium hydroxide. Okay, don't get confused. Then we need standard protein solution. The standard protein solution according to the kit, the Corel kit that we are using is 8 gram per dl. Now, according to various kits, the standard can change. Okay, it can change, it can be 6 as well, it varies kit to kit, but the kit that we are using, the standard is 8 gram per dl. Then we need test serum and distilled water. Then the procedure is same like your Godpot method. You will take 1000 microliter, 1 ml means 1000 microliter. You will take 1000 microliter of the Biuret reagent, and in blank you will take 20 microliter. In Godpot, how much did you take? 10. Here you will take 20 microliter in blank, 20 microliter of distilled water in blank. Then in standard you will again take 1000 microliter of Biuret reagent, and in that you will add 20 microliter of standard. Okay, then in your test you will take 1000 microliter of Biuret reagent, and in that you will add 20 microliter of the serum, that the thing that we will provide to you, the sample. It's the same, only the value has changed from 10 to 20. So in observation, so after you have prepared your blank, then standard and test, what do? You shake, you will mix it, means you will shake the test tubes, and then you will put it, where will you put it? Incubator. For how many minutes? 10 minutes. At what temperature? 37 degree or room temperature. After that you will take it out after 10 minutes and you will take the reading and you will note it down. So this is the calculation formula. Total serum protein concentration in gram per dl equal to ODT, that is OD test, minus ODB, that is OD blank, by ODS, that is OD standard, minus ODB, that is OD blank, into concentration of standard. Okay, it is the same as your Godpot, is it? It is the same thing. Only the thing that will differ here is, in Godpot the concentration was 100, concentration of standard was 100. In here it will be 8, because the kit is having 8. The standard in the kit, the Corel kit that we are using, is 8. If the standard of the kit that we are using, it was 6. If it was 6, then here it would have been 6. Okay, so it depends on the kit that you are using. It depends on the kit that you are using. Okay, don't get confused. Now, do you know the other formula according to the kit? There is another formula. Yes. What is that formula? ODT by ODS into concentration. So did you do using that formula for your glucose? Yes. So see, the thing is the same. If you make the blank 0, and even if you put it in this formula also, the result will be ultimately the same, because if you are putting 0 means what? ODT by ODS into concentration. So ultimately, ghuma fira ke it is the same thing. Okay, you have to discard blank. If you are doing it manually by calculating, doing maths according to by putting it in this formula, then also you have to minus ODB. And if you are doing it with the help of the calorimeter, if you are doing it with the help of the machine, there also you have to minus it. Okay, so ultimately you have to minus ODB. Now whichever way you want. In your Vasudevan, if you are following Vasudevan, in that book you will get this formula. This particular formula you will get, and the ODT by ODS into concentration, standard of concentration standard, that one is according to the Corel kit. Okay, that formula is according to the Corel kit. Okay, don't get confused. And even if some of you are doing the ODT by ODS into concentration, the division 1 if you are doing, there if you get, suppose you got 6, and by applying this formula your friend got 5.8. So is the difference vast or 0.2 difference? 0.2 difference, right? That means the result is same. Okay, The difference is less than 10%, means the result is same. If you give your blood sample in 2 different labs, suppose you want to check your KFT from 2 different labs, you will not get ditto same result. Okay, you will not get ditto same result. Some bit of 0.2, 0.1 difference will be there, because it varies from lab to lab depending on the kit they are using and the formula they are using. So this much minute difference is fine. 0.2 difference means it is the same result, means it is 6 only. 5.8 and 6 is same. Okay, don't get confused, both are right, whichever you want to do and whichever is convenient for you. And the thing in this particular test you say, or the Godpot method, glucose estimation you say, if you are doing, the thing in this particular test is the pipetting. If you are not doing the pipetting properly, that means your test will come wrong. Now if you are supposed to get hypo, suppose you took the sample of hypoproteinemia, okay, suppose the lab made sample you took was hypoprotein. Now in case of hypoprotein, what is the normal range of protein? 6.5 to 8.5. Now maybe the hypo sample, the range should come between 3 or 4. Now if you are getting 1 or in point like 0 point something something, that means your procedure is wrong. Okay, the difference should not come of that much. If the result was supposed to be 3 or 4, a hypoprotein sample lab made, if we provide you an exam, if you got, by doing maths you got 1 or point something, that means your pipetting was wrong, because maths cannot be wrong. The formula is very simple, the procedure is also very simple. The thing that matters is your pipetting, and if you are not doing your pipetting properly, if you are not taking sample properly, means your result will come wrong. Again you have to do it, again another 10 minutes you have to wait. So pipetting should be done very properly, because the amount that you are taking is less than 1 drop. It is less than 20, this microliter, 20 microliter is less than 1 drop. When you will use today, you will understand. So if you are not doing the pipetting properly, means you will get a wrong result. Similar wise, if you get a hyperproteinemia sample, so and if your answer is 20, it should be more than 8.5, but 8.5 means what? Maybe 9, 10. Okay, if you are getting 20 or more than that, means your pipetting was wrong. You have taken more sample. Okay, so pipetting is very important, not just in protein estimation as well as glucose estimation. Okay, are you clear with the formula? Yes. Then these are some important points to note. Other methods of protein estimation, serum protein estimation are Geldal method. K is silent here. K is silent. The pronunciation is Geldal method. And we have another one, Lowry method. So three methods we know. One is Biuret, the one that we will perform. Second one is Geldal, and the third one is Lowry method. Now the causes of hyperproteinemia, dehydration or hypovolemia. Why do we get hyperproteinemia in dehydration? Who can tell me?
If the water content becomes low, then the solid component increases.
Yes.
Concentration.
Yes. If the body fluid is decreased, means the concentration of protein will ultimately increase. Then in chronic infection, why do we get hyperproteinemia? What happens in a chronic infection? Chronic infection means, suppose a patient is bedridden because of sepsis. In infection, he is bedridden. What will happen in the patient's body?
The metabolism will increase.
Huh? Yes. Good. Yes, to fight with the infection. What is gamma globulin? It is a protein. That's why. And as well as in hyperproteinemia also, in this one chronic infection, it can reduce as well. Protein can reduce as well as protein can increase, because in this chronic infection, patient will need protein. So patient will be given more protein, right? So in that, so chronic infection may protein can increase also as well as decrease as well. Then collagen disease. What is collagen? What happens after you get a cut? What will happen after a few days?
Collagen gets exposed.
And then?
Intensive pathway activity.
And then clotting occurs.
Intensive.
How does it look?
Not but sticky for the transparent. Sticky.
After we get a cut, suppose I cut my hand, so after a few days, will I get a scar in there? White scar. Why does that occur? Why does it occur? Why does that white color scar occur? Because of collagen. Some people have very big scars. Some people will have some big scars, some will have smaller. Why does those thick broad scars occur? Because of, yes, because of collagen disease. Okay, because of the collagen disease. Now this is not something very serious. This disease is not very serious. So in collagen disease we can see hyperproteinemia. Then multiple myeloma. Multiple myeloma means disease in the brain. Now you don't have to know much about it because this is a final year topic. This is, you will learn in medicine. For now you just know in multiple myeloma, proteinemia, hyperproteinemia can be present. Then macroglobulinemia. What is macroglobulinemia? What is the meaning? Macro. What is the meaning of macro? Means protein size is more in the blood. Okay. Then we have the causes of hypoproteinemia in liver disease. Then nephrotic syndrome. Why in nephrotic syndrome hypoproteinemia is present?
ग्लोबुलिन और एल्बुमिन की कंसंट्रेशन किडनी में ज़्यादा होती है फिल्ट्रेशन के लिए. अगर कोई भी प्रॉब्लम हो तो यूरिन में प्रोटीन जो आएगा.
हाँ, क्यों आएगा यूरिन में प्रोटीन?
It is not able to
Reabsorb, because your nephrons in nephrotic syndrome, your nephrons are not being able to reabsorb the protein. That's why your proteins will be getting out from your body through the urine, causing hypoproteinemia. What is hemodilutation or overhydration? Excess water. What? So if you have more fluid, then what will happen to the protein? Will it get diluted? Yes. So if there is more fluid, will it get diluted in the body? If the patient is having more fluid in the body, will it get diluted? Will a CKD patient body is having what? A CKD patient's body is having more fluid, yes or no? Is it correct or wrong statement? Yes or no? A CKD patient's body will have more fluid or not? Yes, the patient will be edematous. You know what is edema, right? Yes. The patient will be edematous. So will the protein content in his body normal or less? Less. Why?
Dilution. But in edema, a particular protein should be less than hypoalbuminemia.
So is it protein or not?
Yes.
Yes. So then malabsorption, that is intestinal cause. If your intestine are not being able to reabsorb the proteins, it can ultimately lead to proteinemia, hypoproteinemia. Extensive burns through skin loss. In burns, what happens? If your skin gets burned, the capillaries are exposed, and through the capillaries, the proteins can ooze out. Then A-gamma globulinemia. A-gamma globulinemia is a congenital disease. What is a congenital disease? From birth. So, absence of gamma globulin in the blood from birth is known as A-gamma. Is known as what? A-gamma. Yes. Then malnutrition in Kwashiorkor disease. What is Kwashiorkor disease?
Protein deficiency in babies.
In babies. Is it seen in adults?
No.
No, in babies. Yes. Then causes of increased AG ratio. Hypogammaglobulinemia. Hypogammaglobulinemia means if globulin is less, the AG ratio will be more or less. Don't get confused. If globulin is less, the AG ratio will be more. And if albumin is less, the AG ratio will be less. So causes of increased AG ratio will be in hypogammaglobulinemia, means decreased globulin in the blood. And causes of decreased AG ratio will be in nephrotic syndrome. Why? In nephrotic syndrome, the protein cannot be reabsorbed by the kidney. By the nephrons, it cannot be reabsorbed. So your body is losing. Means albumin is lost. Means albumin is losing. So albumin will be less. Ultimately, the AG ratio will be less. Then multiple myeloma, chronic infection, you can get decreased AG ratio. Okay? Edema. Edema means fluid in the third space of your body. Fluid in the third space. Now, do you know what is the third space? So you just know that if you are having, suppose if your body is not being able to filter out, like in kidney patients, the body is not being able to function properly, so we advise the patient for dialysis, and so if not, the fluid starts to getting stored in the body. Okay, the fluid starts getting stored, then it goes to the feet. Why does it go to the feet? Because it is the most dependent part of our body. The whole body's weight is carried by our feet. Or it can go to the lungs. It can go to the lungs, then that is the pleural effusion that we say, pleural effusion. It can go to the abdomen. Okay, it can go to the abdomen, then in the face, anywhere it can go. So first, to understand edema, you have to first learn what is the third space. Okay. These are all your final year topics. Now you will just note down few words: edema, multiple myeloma. When you will go in final year, that time you will learn in details what is edema, why does it happens, what are the types of edema, how to estimate. There is, so to test edema also there are many. How to test edema? If you press, do you know how to do it? The elasticity, by the elasticity of the skin, you test edema. What degree? First degree, second degree, third degree. So edema is a very vast thing, but for now you just know edema means the excess fluid in the body is edema. Okay. Amlodipine is a drug, hypertensive, antihypertensive drug. Yeah, right? Pressure, yes, pressure oshud. So if some patients are taking amlodipine for prolonged time, like there are 2.5 mg, 5 mg is also there. Now amlodipine, if any patient is taking for prolonged time, as a symptom, the patient can have edema. One of the drawbacks of amlodipine is edema. Yes, pedal edema will be there. Means the fluid cannot pass back. It is not getting distributed to all the parts of your body. It's getting stored in the most dependent portion of the body. So what will you do next? The patient should go and get the KFT done to check whether the kidneys are functioning properly or not. If the KFT is normal, means the problem is with the medication. That means the patient should stop using amlodipine and change to some other composition. Okay, so it's not a disease, it is a symptom. Like fever is not a disease, fever is a symptom of infection in the body. Okay? Biuret reagent. See, Biuret reagent. Everyone can see Biuret? Yes, ma'am. Okay. This is your standard. Do you see 8 gram per dl? Yes. 8 gram per dl, okay? According to the kit. This is the Corel kit. Okay? What is the first group? You and you. Okay, stand. What is the first step you will do?
Biuret reagent.
First you have to take
Biuret लेता है ना?
हाँ, Biuret reagent. Biuret reagent how much? 20 microliter. Yes.
Sorry.
Maths week है.
20. 0.02.
First see, yes, 1000 take.
Already this ना?
Yes. Next. नहीं, गलत. पहले प्रेस करके फिर अंदर लेना. Soft press. Okay. ले लो, done. Press. And we have to activate it for 10 minutes. At 5:30,
answer is done, then we will see for color change. With your eyes, we can see the color change, that is the sample contains. There is no color change, that means there is no protein. That light you will not see any color change. If you have seen some color change, that means your test to have some chemical reaction required. So before you will
Then now you will take standard. That is distilled water. You will
keep on checking. You will see checking. So the limit between like why? Because if there is a bit of fluid over there, it will become diluted. Okay, and your values will also become less. Okay, as compared to what you right? Follow these things when you are prepared. Okay, this is in your mind. Now after you prepare the sample, you are distributed to take it out, and then you take the which is having 10 to 0, but before measuring always cross check. Okay?
mix color.
That is the
Mix and
there is one more process that is reagent time to put the OTP activity Biuret. And that is what we are doing for testing of
और अंदर डालो अंदर नहीं जा रहा जोर से प्रेस करो ओके नहीं प्रेस करके रखो फिर से जोर से प्रेस करो प्रेस करके रखो फिर वैसे बाहर निकालो foam will be there take one red you will take the standard okay who is blank blank there is no problem with that no. You standard हो गया the incubator 10 minutes